In this experiment, we tested diffusion using dialysis tubing, which acts like a cell membrane. For part 1, which is our preparation, we started by putting on safety gears and gloves to protect ourselves. Then we soaked a piece of dialysis tubing in water to make it soft and easy to open. Step 2, setting up the dialysis tubing using tweezers. We carefully opened the tubing and tied one end with a rubber band to create a small pouch. Step 3, which is adding the solutions, we use a pipette to add 3 different solutions into the tubing: 5 milligrams of glucose solution, 7 milligrams of starch solution, and 5 milligrams of distilled water. Then we sealed the other end with a rubber band and weighed the tubing before placing it in the beaker. Step 4, preparing the beaker solution. In the beaker, we mixed distilled water with iodine and stirred it. This was the external solution where the dialysis tubing would be placed. Step 5. Diffusion process. We place the filled dialysis tubing into the beaker and let it sit for 30 minutes to allow diffusion to occur. Step 6. After 30 minutes, we notice color changes. The glucose solution turned yellowish brown. The starch solution turned dark purple blue after reacting with iodine. And the distilled water changed to orange yellow. We also reweighed the tubing to see if its mass changed.
Step number 6. Observations. After 30 minutes, we notice color changes. The glucose solution turned yellow-brown. The starch solution turned dark purple-blue after reacting with iodine. The distilled water changed to orange-yellow. We also reweighed the tubing to see if its mass changed. Step 7. Testing for diffusion. To check what diffuse, we use glucose test strips and compare the results. Glucose was found inside and outside the tubing, meaning it diffused. Starch stayed inside because its molecules were too large to pass through. Iodine diffused into the tubing, reacting with the starch. Step 8. Conclusion. Our results show that smaller molecules like glucose and iodine can pass through the membrane, but larger molecules like starch cannot. This models how real cell membrane works in living organisms. Step 9. Cleanup. Finally, we put away the dialysis tubing, washing the glassware, and clean up our workplace.